In October 2013, a paper published in Science by Lulu Xie, Hongyi Kang, Maiken Nedergaard, and colleagues at the University of Rochester reported something that had not been formally demonstrated before. The living brain, during sleep, dramatically increases the exchange of cerebrospinal fluid with interstitial fluid in brain tissue, flushing out metabolic waste products including beta-amyloid, the protein that accumulates in the plaques found in Alzheimer's disease. The researchers measured this in mice and showed that during slow-wave sleep, the interstitial space in the brain expanded by approximately 60%, allowing cerebrospinal fluid to move much more freely through the tissue. This observation, of a dedicated waste clearance system operating primarily during deep sleep, has been cited over 5,000 times and fundamentally shaped how neuroscientists think about the relationship between sleep and Alzheimer's risk. In this video, what the glymphatic system is and how Xie et al. Demonstrated it, what the 60% figure actually means mechanistically, how this connects to human data on sleep and Alzheimer's risk, what the critical limitations of this research are before drawing clinical conclusions, and 3 takeaways for adults over 55 who want to optimize their sleep based on current evidence. Quick note before we continue, this is educational content based on published research, not medical advice. Talk to your physician before changing your regimen. Now, the study. Before 2013, the prevailing understanding was that the brain lacked a conventional lymphatic drainage system, unlike most other organs in the body. Peripheral organs use the lymphatic system to clear cellular waste, immune cells, and excess fluid. The brain, protected by the blood-brain barrier, was considered to manage waste through slower mechanisms including diffusion and active transport across the barrier itself. The Nedergaard laboratory had published preliminary work in 2012 describing a system of fluid channels along blood vessels in the brain, which they termed the glymphatic system, for its glia-dependent function. The 2013 science paper was the direct experimental test of whether this system was functionally active during sleep and whether its activity was relevant to the clearance of pathological proteins. The experimental model used was in vivo 2-photon microscopy in anesthetized and awake mice. This is a high-resolution imaging technique that allows researchers to visualize fluid movement in real time in living brain tissue. The researchers injected fluorescently labeled tracers into the cerebrospinal fluid of mice in different states, awake, anesthetized to simulate sleep, or naturally sleeping. They then measured the rate at which these tracers penetrated brain interstitial tissue and the speed at which injected beta-amyloid was cleared from the brain. The comparison between awake and sleep states was the critical experimental variable. Here are the results. During sleep, the interstitial space in the cerebral cortex expanded by approximately 60% compared to the awake state. This expansion was associated with dramatically increased convective flow of cerebrospinal fluid through the tissue. The rate of beta-amyloid clearance from the brain was significantly faster during sleep than during wakefulness. The researchers estimated that beta-amyloid clearance was approximately 2-fold faster during sleep. Critically, when they used a pharmacological agent to reduce aquaporin 4 function, aquaporin 4 is the water channel protein on astrocyte in feet that mediates glymphatic flow, they substantially impaired this sleep-dependent clearance. This mechanistic intervention linked the structural observation to a functional outcome. The study also tested whether this effect was specific to natural sleep versus anesthesia-induced unconsciousness. The glymphatic activation was observed in both anesthetized animals and naturally sleeping animals, suggesting it is a property of the low arousal, slow-wave-dominated brain state rather than a consequence of anesthesia chemistry. The slow-wave brain state is characterized by synchronized, high-amplitude, low-frequency electrical activity across the cortex, what sleep medicine refers to as stage 3 sleep, or slow-wave sleep, in the human 4-stage classification. It is the deepest non-REM sleep stage. In humans, slow-wave sleep is most abundant in the first half of the night and declines significantly with age. How does this connect to human data? The mouse findings stimulated a wave of human neuroimaging studies. A notable example, a 2017 paper by Lulu Xie and colleagues in Brain, and subsequent work by Lulu Xie, and Holtzman at Washington University, used cerebrospinal fluid sampling and PET imaging to show that even a single night of sleep deprivation increased beta-amyloid burden in specific brain regions in healthy middle-aged adults. The effect was detectable after just one night. A 2019 paper by Lulu Xie and colleagues published in Science Translational Medicine found that reduced slow-wave sleep, not total sleep duration, was the specific sleep metric most strongly associated with higher cerebrospinal fluid levels of both beta-amyloid and tau, the two pathological proteins that define Alzheimer's disease neuropathology. This is a crucial distinction for people over 55. It is not just about sleeping 8 hours. It is about the quality and architecture of that sleep, specifically the proportion of slow-wave sleep. Slow-wave sleep decreases substantially with age. Studies using polysomnography show that adults in their 60s and 70s typically spend 50 to 75% less time in stage 3 sleep than adults in their 20s. This age-related decline in slow-wave sleep happens independently of total sleep time. A person sleeping 7 hours at age 65 may have dramatically less slow-wave content than they did sleeping the same 7 hours at 35. Now for the critical limitations. First and most important, the Xie et al. 2013 study was conducted entirely in mice. Translating findings from rodent brain physiology to human pathophysiology is not straightforward. Rodent and human brains differ substantially in size, architecture, and the proportional roles of different sleep stages. The glymphatic system has since been identified in human brain tissue using post-mortem studies and MRI-based imaging techniques, which is supportive of the basic mechanism. But the direct quantitative measurements of clearance rates during human slow-wave sleep have not been replicated with the same precision as the original mouse experiments. Second, the observational human studies showing links between poor sleep and Alzheimer's risk are largely correlational. Poor sleep could be an early symptom of Alzheimer's pathology beginning to disrupt sleep-regulating brain structures rather than a cause. The direction of causality in humans remains an open question that longitudinal studies are actively working to clarify. Third, most human sleep intervention studies are short in duration. Whether improving slow-wave sleep architecture over years measurably reduces beta-amyloid accumulation in living human brains is not yet established in long-term randomized trials. The mechanistic case is strong. The causal chain from sleep improvement to reduced Alzheimer's risk in humans is plausible but not yet proven. A fourth limitation worth noting: the glymphatic system is not the only mechanism by which sleep affects brain health. Sleep consolidates memory through hippocampal replay processes, regulates cortisol and stress hormones, and supports synaptic pruning, all of which have independent relevance to cognitive aging. The glymphatic story is compelling and well supported, but attributing all of sleep's cognitive protection to one mechanism oversimplifies the biology. 3 takeaways from this evidence. First, slow-wave sleep is the metric worth paying attention to, not just total sleep duration. If you use a consumer wearable with sleep staging, looking at your stage 3 or deep sleep proportion is more relevant to the glymphatic hypothesis than total hours. Most commercial devices that claim to measure slow-wave sleep use motion sensors and heart rate, not EEG, so treat the numbers as rough approximations. A sleep specialist can conduct polysomnography if you have specific concerns about your sleep architecture. Discuss with your physician whether a sleep evaluation is appropriate for your situation. Second, the factors that most reliably increase slow-wave sleep proportion in the research literature include consistent sleep-wake timing, limiting alcohol in the hours before sleep, which research has shown suppresses slow-wave sleep in the first half of the night despite often helping initiation, physical activity during the day, and avoiding large meals close to bedtime. None of these are medical prescriptions. They are variables that appear in the sleep architecture literature with reasonable consistency. Whether they apply to your specific situation, particularly given any medications you take or conditions you manage, is a conversation for your physician. Third, beta-amyloid and tau levels are measurable in living people through cerebrospinal fluid sampling and PET imaging, though these are research and diagnostic procedures rather than routine checkups. Blood-based biomarkers for amyloid, specifically plasma amyloid beta-42 to amyloid beta-40 ratio and plasma phosphorylated tau, are becoming more available in specialized clinical settings. If you have a family history of Alzheimer's disease or are already working with a neurologist for another reason, asking whether any biomarker assessment is appropriate for your profile is a legitimate clinical question grounded in current science. To summarize, The Xie et al. 2013 science paper demonstrated in mice that the brain's glymphatic system expands interstitial space by approximately 60% during sleep and clears beta-amyloid approximately twice as fast during sleep as during wakefulness. Subsequent human studies have linked reduced slow-wave sleep specifically, not just total sleep duration, to higher cerebrospinal fluid levels of Alzheimer's-associated proteins. The causal link in humans requires further long-term evidence. This is educational content, not medical advice. Always consult your physician before changing your regimen. Studies are preliminary, individual responses vary. If you found this useful, our free guide, How to Read Your Biomarker Panel at 55+, includes a section on cognitive aging markers. Link is in the description. New evidence reviews publish every Tuesday and Friday.